Thank you to the organizers and also to the three wonderful speakers before this session and then also the keynote. So you've gotten a sense of maybe how theoretical physicists view this range of problems, and over the next half hour I'll give you a sense of at least how this experimental physicist looks at these questions of tipping points. And in particular, my group in the physics department over at MIT, we're interested in using laboratory microcosms, so laboratory microbial populations that we can experimentally kind of turn knobs and make quantitative measurements in order to understand really this broad question of how interactions between individuals in a population can really dictate both the evolutionary and the ecological dynamics of that population. Right? Now, as you've heard, there are many examples in which natural populations can collapse suddenly in response to deteriorating environmental conditions. And what's interesting is that from ideas from nonlinear dynamics, there are at least in principle universal behaviors of complex systems near such tipping points that may allow us to, in principle, anticipate in advance that the transition is about to take place and perhaps intervene to save the population. And in particular, what we've been able to measure experimentally is that there really is a change in the fluctuations of a population before one of these tipping points. Now, the basic reason that these populations are subject to a sudden collapse is because there's these positive feedback loops that are stabilizing the state. And in particular, in my yeast populations, they're collectively breaking down the sugar sucrose. And now, anytime you have such a collective behavior, you have to ask, well, what happens if one member of the population stops contributing to that public good? All right, so if you wanted to, you could call that individual a cheater. All right? But the point is that such a cheater strategy may be able to both emerge and spread in the population. And in that case, this original state of cooperation may be unstable in some evolutionary sense. And indeed, in our experimental populations, what we find is that such a cheating or non-producing strain of yeast can indeed spread in the population. Right? However, we find that there's not necessarily immediate collapse resulting from that, but rather we end up getting coexistence between both the cooperators and the cheaters, even in these very simple, kind of well-mixed environments that we're performing these experiments. Despite the fact that we get coexistence, we still observe a variety of what you might call deleterious consequences associated with the cheaters. In particular, there's a loss of resilience in these populations. All right, now, I guess we've brought up this question of fisheries and fishery collapse, and I just want to show you some of this data from the Newfoundland cod fishery. Right, so on the y-axis is, unfortunately, not the population size, which is what you would like to plot, but it's instead the number of fish that are caught as a function of time. Right, and what you can see is that this was a very productive fishery for really hundreds of years. However, in the 60s and 70s, improved fishing technology led to this dramatic increase in the number of fish that were caught. And eventually, this increase in fishing led to a catastrophic collapse of this fishery. So although we haven't been fishing in this region for 20 years, only now are there tentative signs of recovery. Okay. Now, the basic reason that these populations can exhibit such sudden transitions is because of the positive feedback loops, as I mentioned. Right, so in the context of fish, there might be schooling behaviors, land animals herd. There are many examples of collective hunting type behaviors, for example, how wolves can take down large animals. And more broadly, animals need to find mates. So below some density, the population may be in trouble. Right? Right. Although these sorts of collective dynamics take place in the macro scale, they also take place at the micro scale. And I'll tell you about an example in my yeast populations. Okay, so we've mentioned kind of, there have been a number of these plots that have come up that are these bifurcation diagrams, and we haven't really walked through it in too much depth. So I'll give a concrete example in a moment, but the general cartoon is that what can happen is that if you look at the size of a population as a function of some environmental condition with deterioration on your right, what can happen is that as the environment deteriorates, there's a modest decrease in the size of the population, but perhaps nothing that you would say is especially alarming. However, at some critical level of the environment, so some level of fishing, for example, there can be this catastrophic collapse. Could be local extinction or just a transition to some other, we'll say, undesirable state. Okay. However, from here, what you can ask, well, what happens if we improve the quality of the environment? And it's actually, it may be that you have to come all the way over here to this point on the left before you get a transition back to the original state. Okay. So what you see is that at this intermediate environmental conditions, the system is actually bistable. Okay. So there are these two stable states that are separated by some unstable state that's denoted by this dashed line. All right, so that's telling us about kind of the minimal size required for survival. Right, so if you start out above it, you'll come up, but below it, you'll come down. Right, so this is what's known as a fold bifurcation because those fixed points are folding over on each other. And it's the basic mechanism that is thought to lead to sudden transitions in a wide variety of these complex systems that you've been hearing about earlier today. Okay. Now, of course, in a lot of these situations, what you'd really like is some way to anticipate that you're approaching that tipping point before you cross it, because after you cross, it may be difficult to get recovery. Okay? Now, the interesting thing is that from nonlinear dynamics, there are predictions that there are, at least in principle, universal behaviors of populations, ecosystems, and other dynamical systems as we approach such a bifurcation. Right? And to get an idea of why that is, what we can do is we can take this bifurcation diagram and we can take two slices of it. Right? So this green slice is kind of far from the tipping point, F2, and the red slice is when we're close to it. Right? Now what we're going to do is we're going to rotate things. Right? We're going to plot, now it's the population size on the x-axis, and there's an effective potential here on the y-axis that is telling us about how the population will respond to perturbations. The idea here is that if we're far from the tipping point, then this stable state here is a very nice potential well. Right? In particular, any perturbation away from equilibrium will quickly go away. And also, you can see that there's a very large, what we call basin of attraction. Right? So a very large perturbation will be required in order to knock this system into the alternative state, in order to cause collapse or extinction of that population. Okay? However, as we move close to that tipping point, what we see is that the effective potential looks very different. All right? So now, it still is a stable state, but somehow it's not as stable. So there's a loss of stability. The shape of the potential is much broader. And this tells us any perturbation away from equilibrium will take longer to go away. And also, even in the absence of a defined perturbation, just the natural fluctuations or noise in the system will kind of excite the population, and you expect then a change in the fluctuations of the population. The expectation is that the fluctuations should get both larger and slower. Finally, what you can see is that this basin of attraction has now shrunk. Right? This is telling us that a smaller perturbation here could cause transition to the alternative state. Okay? The mathematical ideas behind this, it's known as critical slowing down, and it emerges from the fact that this is a bifurcation in which the eigenvalue goes to 0. All right, and it's an important statement that even in high-dimensional systems that cannot be described as an effective potential like this, nonetheless, in this kind of bifurcation, you expect the dominant eigenvalue to go to 0. So in some sense, this critical slowing down phenomenon is even more general than what you'd expect from the simple kind of cartoons that we typically imagine in the context of this potential picture. Okay? All right, so if you've kind of gotten lost of what the predictions are from the theory, I just want to highlight their general expectations of these complex systems as they approach a tipping point. That can be summed up as a few different things. So first, you expect there should be a loss of resilience to environmental perturbations as you approach one of these tipping points. And that this loss of resilience should be accompanied by some possible early warning signals. Right? So first, what you should expect is there should be an increase in the time to recover from perturbations. And second, there should be an increase in both the size and the timescale of fluctuations in the system. Okay? All right, so from my standpoint as an experimentalist, I think that these are very interesting theoretical predictions, but we want to know, can these things be measured in any real living population? All right, so this is the goal that two students in my group set for themselves. And what they decided to do is to use just laboratory yeast populations that have been studied in many different contexts by biologists. All right, so these are single-celled organisms. They're just a few microns in size. Right? Associated with their small size, they're relatively simple with a short generation time. Right? They can divide every couple of hours. Moreover, we can perform very quantitative measurements of things like the size of the population, and we can directly manipulate the environmental quality. Right? So we have very clear experimental knobs where we can control things like just how challenging the environment is. And then finally, we can also perform various genetic manipulations that allow us to control the strategies that are being followed by those yeast cells. Okay. All right, so what you need to know about the yeast right now is just when they're growing on the sugar sucrose, so this is common table sugar, right, what you put in your coffee this morning. Right, the way that they grow on this sucrose is that they secrete an enzyme outside of the cell. Right? And this leads to some positive interactions between the members of the population. And in particular, in a very concrete sense, what you can measure is that the yeast divide more rapidly at higher cell density. Okay? Of course, eventually, they'll run out of resources and the rate of division will go back down. But over some range of densities, the yeast benefit from the other yeast in the population. Okay? And it's those positive feedback loops that lead to this possibility of sudden collapse. All right, of course, we want to go and try to explore this experimentally. All right, our experiments here are really rather simple. We're growing the yeast in what microbiologists call serial batch culture. All right, what this means is that we start at the beginning of the day with a low density of these yeast cells, and we let them divide over the course of the day. Now, at the end of the day, we can just measure how many cells there are by looking to see how cloudy that liquid is, the optical density. Okay? And then we just dilute by some amount and transfer a small fraction of the cells into fresh media where we supply fresh sucrose and all the other nutrients they need for growth. Okay? Now, the data I'll show you today, what we've been doing is controlling the quality of the environment by varying this dilution factor. Right? So a larger dilution corresponds to a higher effective mortality rate, and hence a harsher environment. Right? And so for example, we're going to start by looking at what happens if we dilute by 1,400 each day. Okay? And the first thing we wanted to ask is it really the case that these yeast populations, when growing in sucrose, display bistable fates depending upon the starting density of the cells? Because this notion of bistability is really central to the situations that should lead to sudden collapse. Right, and this is the kind of thing that is easy to do with yeast, difficult to do with fisheries. We just start populations at many different densities, and then we just propagate them, daily diluting each over time, and measure the size of the populations at the end of each day. What you see is that if we start with a large density of cells, those populations are all stable, they survive. However, if we start with a low density of cells, those populations, they shrink over time, and indeed they go extinct, and that's another stable state. Okay? Now, the expectation is that between those two stable states, we should have an unstable state. Right? And indeed, you can see really where that is. And these 8 populations, although nominally identical, seem to experience stochastic fates. Some of them look like they're going to survive, and some of them are going to go extinct. And this is basically because we placed those 8 populations at the top of some effective potential. So some of the populations are falling to one side, and some to the other. Okay? Right? It's important to note here that it's not that those cells, that they were unable to survive or unable to divide, but rather there were not enough cells to break down enough of the sugar to allow them to divide enough to counteract this just daily death process, the dilution that we're doing each day. Okay? All right, so from this sort of data, you can see kind of the locations of these various fixed points of the system, and we can just plot them. Right? We can look at, this is the stable size, the minimal size required for survival, and again, there's another stable state down at 0. And this is all at a particular environmental quality, a dilution factor of 1,400. Okay? But the reason that we started these experiments is because we wanted to explore this idea of a fold bifurcation. We want to ask, would this population really experience one of these critical transitions leading to a catastrophic collapse? Right? In order to get at that, what Ley did was just repeat this experiment, but for many different dilution factors. And what he found is what I think is really a beautiful fold bifurcation describing how normal laboratory yeasts grow on this simple sugar sucrose. What you can see is that the size of the population indeed shrinks as the environment deteriorates, and the minimal size of the population required for survival, that grows. And indeed, this tipping point occurs when the minimal size of the population is equal to the actual size, and that's when you get this bifurcation leading to collapse. Okay? Now, from our standpoint as experimentalists, if we want to measure behaviors of at least this population near a tipping point, it was really essential that we start by confirming that there really was one of these bifurcations, a tipping point. Because in this field, many researchers had models saying that a particular system should perhaps have such a fold bifurcation, but this was the first time that it was really just possible to just experimentally go in there, turn all these knobs, and make direct measurements to confirm that that's what was going on. Okay? And now that we have this, we can go and we can test all these kind of classic ideas from theoretical ecology. All right, so for example, this idea of the resilience of the population can be thought of in some ways as the distance from the stable state to the unstable state. Because if you're over here on the left, a particular perturbation may push the population here, in which case the population will recover to its original state. However, a population over here, when subject to the same environmental perturbation, could be pushed below that unstable state, and then you get collapse of the population. Right? So this is kind of why you expect a loss of resilience. Right? But in the laboratory, we can go and just see this directly. Right? What we did is we allowed populations to reach equilibrium over the course of 4 days, right? So roughly, say, 40 generations. And then for 1 day, we just expose them to a mild salt shock, right? That inhibits growth in some way. And then we return the population to the original environment. Okay? What you can see is that in a benign condition, low dilution factor, the population dips in size, but you get rapid recovery. However, as the environment deteriorates, what you see is now there's a larger dip, and it's taking longer to get recovery after that perturbation goes away. So we can already directly see this basic feature of critical slowing down. It's taking longer to recover from a perturbation as the environment deteriorates. Okay? In a more challenging environment, now what you can see is that some of the populations may survive and some may not. And in a yet more challenging environment, those populations are all going extinct. Okay? So this is populations subject to the same perturbation, but some of the populations are surviving and some are not, depending upon where they are on this bifurcation diagram. Now, it's worth mentioning that Martin Skeffer, who has been one of the prime motivators in this field, he used another microbial system in continuous culture, so not with this daily dilution, but just in the equivalent of a chemostat, and he was able to measure an increase in this recovery time there as well. Right, so there's no sense that the data that we're seeing here is specific to this daily dilution. This is just one way of implementing the experiments. All right, so now we can come back to this bifurcation diagram and ask about these fluctuations. All right, so it's expected that as we move to the right, that the fluctuations in the system should grow. Okay? All right, in order to look at that, what we can do is we can look at the coefficient of variation. So this is the size of the fluctuations in the population size over time, right, over a replicate of 20 to 30 different populations, okay, as a function of the environmental condition. What you can see is that in benign conditions, the fluctuations are only a couple percent, so very small. However, as we approach this tipping point over here, what you see is there's quite a dramatic increase in the size of the fluctuations. Right? And indeed, even if you look at just the raw magnitude of the fluctuations, even that's growing as we approach this tipping point. Right? So this is something that you would very much not expect from any sort of demographic fluctuations. It's really a rather specific indicator of this critical slowing down phenomenon. Right? I'm not going to show you the data, but we also measured the autocorrelation time, so the typical time scale of the fluctuations, and we found that they went from 2 days to 7 or 8 days. Right? So the fluctuations not only became larger, but they also became slower as we approach this tipping point, as predicted by theory. Okay? And this is telling us that we could really use just the fluctuations in these populations as an early warning indicator to tell us that we're approaching this tipping point. So in principle, we could have, written to Congress to intervene to save these yeast populations. Okay? So one of the things that we find exciting about these notions of critical slowing down is that they're nominally universal, and that they may show up both in our yeast populations, but also maybe in fisheries. But also, in a specific system, they should show up under different forms of environmental deterioration. Right? And in a set of experiments I'm not going to have time to tell you about, we've been able to measure this change in behavior under 3 different forms of environmental deterioration. So not only this one, but decreasing the sugar concentration and also changing, for example, the salt concentration. And we can measure these sorts of indicators before collapse. Right? So this is giving us some measure of comfort that these things have a degree of universality, at least. Now, in all the experiments that I just told you about, we were studying kind of isolated yeast populations where there was no notion of any spatial degree of freedom. However, real populations are distributed over space, and so it's natural to ask what are the typical kind of spatial indicators of an impending transition. And in this paper, what we've been trying to do is think about whether there might be what you might call the spatial analog of recovery time. Okay? So the way that I like to think about these indicators is by looking at whether we're kind of measuring the behavior of the system as a function of time or as a function of space, position, and also whether there's a perturbation or not. Okay? So the basic phenomenon of critical slowing down is telling us that as we approach this tipping point, it takes longer for a population to recover from a perturbation, or a population or other complex system. Okay? Now, we were able to measure this increase in recovery time in our yeast populations. We also were able to measure this other signature, which is that even in a uniform environment, so constant over time, nonetheless, you could look at the fluctuations in the size of the population to tell you whether you're approaching a tipping point. And indeed, we were able to measure that. Okay, now, if we think about the spatial populations, well, you could think about an environment that is uniform over space, and then the corresponding thing you would look at is density fluctuations. And this is familiar to many people in the context of phase transitions. What was surprising to us is that in this field of early warning indicators, nobody, as far as we could tell, had proposed what is a rather natural fourth category of indicator. Right? So this would correspond to the spatial analog of recovery time. Right? The idea here is that if you have a boundary between 2 regions of different quality, right, then you have to get some distance away from a poor region before you get to your equilibrium density. Okay? And that's just because there's some depletion effect where individuals over generations and so forth are kind of moving between these different regions, so you kind of get a depletion of your density as you approach a region of poor quality. Right? The nice thing about this recovery length is that it's a deterministic quantity that can be measured at a particular moment in time. So you don't need a high-quality time series. You don't need to have a defined perturbation in time like a drought. Instead, you just look for a boundary between regions of different quality, and then you measure the density profile approaching that boundary. Okay? And indeed, through some combination of theory, computation, and experiments in the lab, we were able to show that this is really a rather powerful indicator that can be measured quite cleanly. And consistent with this, we've recently been collaborating with a professor at the University of Pisa, Lisandro Benedetti-Cecchi. And what he was what he was able to demonstrate is that in an intertidal community, so this is an actual, real, honest-to-goodness ecosystem on an island in the Mediterranean off the coast of the University of Pisa that has alternative stable states between a canopy state and an algal turf state, that by going in and manipulating these ecosystems directly, we were able to measure an increase in this recovery length in such a real-world setting, which is giving us some confidence that this may be a useful early warning indicator. Now, I'm not going to get into this very much, but once again, we're excited about these sorts of tipping point ideas because we think that they may be relevant across different scales. And in addition to this being kind of the basis of sudden collapses in populations, it's also the basis of cell memory. Right? The idea is that if instead of the axes that we had before, now we can think about some phenotypic state of a cell as a function of an inducer concentration, you can see that this thing is representing a memory module of some sort, because if you're at this intermediate environmental condition, the cell is retaining memory of its past exposure to some external inducer. Okay? And what we've been doing in experiments in my group recently is taking some canonical examples of such a memory module, such as this toggle switch in yeast. Right? And this is from the Collins lab. This is not the original toggle switch, because this one's in E. coli. Or sorry, the original one was in E. coli. This one's in yeast, but it's the same basic design, where you have mutual repression of two different transcription factors. Right? And this is a very nice system because there are two different fluorescent proteins, so we can really see what's going on. We also have two different small molecules that we can use to control the strength of the repression. Right? And consistent with other measurements, this system has very well-defined memory, in the sense that if we start the system in this high GFP state, then we let them grow, in this case for 100 generations. What we see is that the cells retain memory high, until all of a sudden they switch to this low state. Okay? But this thing really is a fold bifurcation, because you can show that if you start here, then it retains memory of this lower state. Okay? Now the question is, well, what happens if we look at these populations over here? In particular, what we find is that the rate of stochastic switching is really very low. Right? It may be that after 100 generations, only 1 in 1,000 cells have switched. But nonetheless, what we see is there's a loss of resilience to perturbations. Because what we can do is we can perturb the cells with a wide variety of different environmental perturbations. Right? Whether it's salt or temperature, pH, a variety of different perturbations can actually cause loss of cell memory, loss of memory of this phenotypic state, and induce switching to this low state down here. And we think that this may be a rather general phenomenon. And indeed, we've been able to see this in this synthetic circuit as well as natural circuits in both yeast and in bacteria. All right, but I want to kind of end by coming back to this question of the collective behavior of the yeast populations. Right? So we have a population of yeast. They're all kind of happily growing on this sugar sucrose. But then we have to ask, well, what happens if one of those cells stops contributing to that public good? Right? If you'd like, we could call it a cheater, or if you prefer, you could call it a non-producer. Whatever you call it, it doesn't change the results of the experiment. Right? And then what you have to ask is, well, if the only difference between these cell types is that the cheater doesn't have to pay the metabolic cost associated with making and secreting that enzyme, well, in that case, what you'd expect is the cheater should be able to spread relative to the cooperator. And indeed, in a pure kind of public good type interaction, the cheater always has an advantage relative to the cooperator, and that should lead to extinction of cooperation. Okay? And that's the basic outcome that is expected from some of these classic models from evolutionary game theory. Right? But we're experimentalists, so what we did is we just took these normal wild-type yeast cells, and we competed them against a knockout. Right? So this is just a strain of yeast where the gene required for the cooperative behavior has been removed. Okay? And we also put in different colored fluorescent proteins so that we could track the dynamics by flow cytometry. Okay? Now, what we're going to do then is we're going to start, say, with 1% cheater in a population of cooperators and ask, well, how is it the cooperator fraction changes over time? And what we found experimentally is that, indeed, the cooperator fraction is decreasing. Right? So this is telling us that that cheater strain can indeed take advantage of the cooperators and spread. Right? Now, in general, as this occurs, we observe a decrease in the growth rate of the population. Right? So this is a very concrete case in which evolution leads to a decrease in the fitness of a population. Right? And this is something that can generically happen when you have interactions between different cell types. Now, this decrease in cooperation is consistent with this basic notion of a public good type dilemma, in which maybe it's just cooperation in this well-mixed environment is just not evolutionarily, it's not sustainable. Right? But to get at that, we have to look not only to see this, but we have to start at low cooperator fraction and then see what happens. Okay? And when we did that experiment, what we found is that if you start with 1% cooperator, 99% cheater, then it's actually now the cooperators that have the advantage. Right, so in this case, what we see is that there's what we know as negative frequency-dependent selection. Right, so rare strategies are somehow doing better than common strategies. And this leads to coexistence between the two types. And indeed, we can measure the equilibrium fraction as a variety of different environmental knobs and so forth, but the key feature is that really this coexistence is a very robust feature of this system. Okay? And it's something that we've seen not only in yeast, but in a variety of other kind of public good type interactions. Okay? Of course, you have to ask, well, how is it that those cooperators are able to survive the presence of the cheaters? Okay? And what we found is that if you look at the cooperators, they indeed make this enzyme called invertase, and they secrete it outside of the cell. But some fraction of those enzymes get stuck in the cell wall of the yeast. Right? So it's secreted out of the plasma membrane, but some of it gets stuck in the cell wall. What that means is that when all is said and done, some of the enzyme is nearby, so that means that some of the public good is created locally. That gives the cooperator cells some preferential access to the fruits of their labor. Right? It allows them to survive in the presence of those cheater cells. Now, a significant kind of focus of the group over the last few years has been trying to understand the various maybe ecological forces that may either help or hinder the evolution of cooperation within a species. And I'll just mention, for example, one thing that we found that was perhaps a bit surprising is that if you take another species, such as, say, E. coli, and you add them to the same test tube as the yeast, this actually drives cooperation within the yeast population. Right? So there's a sense in which competition between species can be a stabilizing force for cooperation within a species. And if you're curious about these sorts of things, you can ask me later. Okay? But here at the end, I wanted to just say something about, in this system, there's very interesting couplings between the evolution and the ecology, between the evolutionary dynamics and the population dynamics. Okay? And this is a situation where this coupling is perhaps surprising, and that's just because we typically assume that there's a separation of time scales between evolution, which is very slow, and ecology or population dynamics, which are rather rapid. Right? So what that means is that if you want to understand the dynamics of humans on Easter Island, you don't need to try to think about maybe evolutionary changes in the state of that population, but that's because of this separation of timescales. However, in a number of different systems, recently ecologists have gotten at least some evidence that these timescales can overlap, in which case that you can get feedback between these two processes. And this might be especially prevalent in microbial populations because evolution can be quite rapid as a result of strong selection. Okay? All right, so coming back to if you look at pure cooperator populations, which I started out talking about, then the population dynamics are relatively simple. Right? So there's some minimal size required for survival, otherwise they go extinct. Okay, but now there's a question, well, what happens if we look at the changes in the size of mixed cooperator-cheater populations? All right, well, in that case, what you see is that things actually get quite tricky. Okay, all right, so what happens is that population sizes can go down and then up, then down. So it's actually kind of difficult to figure out what's going on. And indeed, even if you track the evolutionary dynamics of this population over time, again, you see things coming up and down, and it's really rather a mess. Okay, so it's easy to look at this sort of data and just kind of throw up your hands and say, oh, biology, it's too hard, and go back to studying electrons in carbon nanotubes. Okay? But that would be a mistake, because if you take this precise data, and instead of plotting them relative to time, if you instead plot them relative to each other, then you start to see some, I think, quite interesting patterns. What you see is that this red population is indeed going extinct, but as that happens, it's actually becoming more and more cooperative. Whereas these other populations are doing some sort of interesting spiraling over time. And each arrow corresponds to the change in the size of the population and the population composition over the course of one day. Right, so what's happening here is that if you don't have very many cooperators, there's not very much food, so the population shrinks. But as that occurs, then you reach this regime where the cooperators have an advantage relative to the cheaters because of that preferential access that they had. Right, so in that case, the cooperative fraction grows. But when that happens, okay, now you have a lot of cooperators, so they're creating a lot of sugar, so population size can grow. But once you're here, this is the situation where the cheaters have a clear advantage over the cooperators, so then you get a decrease in the cooperative fraction over time. Right, so there's some explicit coupling between this ecological process and the evolutionary process, so this eco-evolutionary feedback, that is something we can directly see in these spirals that are observable in our populations. Indeed, a very simple model suggests that the dynamics should basically look like this, where populations to the left go extinct, to the right there should be some spiraling to a state of coexistence. And indeed, if we start at many different population sizes and compositions, this is basically what we see. These populations all eventually go extinct. These populations spiral to this red state here. Okay? Now, it's interesting to note that the equilibrium size of the population is not significantly decreased as a result of the presence of those cheaters. However, what we see is there is a loss of resilience, because resilience is still the distance between the state and that separatrix. Right? So, and this is something we could also see experimentally by just shocking the cells, the populations, and seeing if they survive. Okay? Right, now, if you zoom in on that fixed point, what you see are there are really quite beautiful spirals to a state of coexistence. Right, so this is the experimental measurements, changes in each day, where we're all kind of coming into here. And a natural question to kind of bring things full circle is to ask, how is it that these spirals change as we approach the point of collapse? So as the environment deteriorates, how is it that this couple dynamic changes? And what we found is that the spirals become more spirally. All right? And the mathematical statement is that, well, because we have a spiral, now there are two eigenvalues, and they're complex conjugates of each other. And the expectation of what we were able to show is that it's effectively the real part of the eigenvalue that then goes to 0. Right? And now I'm going to just show you what this looks like in the context of a simple model, so you can kind of visualize what we think is going on, which is that in a benign condition, you get rapid spirals to this state of coexistence. But as the environment deteriorates, the spirals become, as I said, more spirally, until eventually you basically get neutrally stable orbits, and then you're going to get collapse of the population. Okay? All right, with that, I'll go ahead and close. All right, so what we've been doing in these experiments is, in a very kind of tractable system, we've been exploring this phenomenon of catastrophic collapse, where we can experimentally map out this fold bifurcation and measure some of these universal behaviors that are predicted by theory. Right? We've also found that experimentally, such populations are often susceptible to cheating strategies, but in a wide variety of contexts, we've observed coexistence between cooperators and cheaters. And we've also seen this in the context of, for example, antibiotic resistance, where resistant cells can, in some cases, protect sensitive cells, and we end up getting, again, coexistence. More broadly, in my group, we're excited about the possibility of moving towards real multi-species communities, where we can take this sort of kind of quantitative experimental approach to try to understand some of the universal rules of community assembly. We're studying this in the context of test tubes, as well as in what I think of as biological test tubes, which is the gut of the worm C. elegans. Right? And with that, I will thank the group, who are the ones that have been doing the work. In particular, Lei, Kirill, and Al did much of the work that I talked to you about today, and also various funding agencies. The New Innovator Award funded the work on tipping points, and the Allen Foundation has funded a lot of the work on cooperation. And with that, I'll thank you.